Good morning, grade 12 students. In this session, we're going to continue with our immunology chapters. I'm here with document 7. Let's talk about the immunological memory on your book on page 151. And this session, we're going to talk about the characteristics of the immunological memory. We're going to talk about the primary and about the secondary immune response. Already we talk about this in the introduction of the immune system. So we're going to move about the immunological memory. So one of the most remarkable effects of the specific immunity is the establishment of a state of long-term resistance. What the meaning of long-term resistance is the resistance for a long period of time against any antigen after it has been encountered for the first time. And for example, if I get flu virus, I encounter this antigen of the flu virus for the first time. So this will create a long-term resistance against this antigen. If this antigen is encountered for the second time, this will increase the chance of eliminating it after a short period of time. So the immune system, it keeps in memory the encounter with the antigen and sends a counter attack against it. And it means a counter attack. Counter attack, in football it's called counter attack. هجمة مرتدة. So counter attack against it, they attack it immediately whenever it enters the body. So this flu virus بيكون فايت in the body. He's thought that he's going to work and going to act as much as he can, and he can do whatever it wants. So the immune system created a memory against this flu virus or the antigen of the flu virus. So it will create a counter against it. So how does the immune system keep the antigen in memory? What are the characteristics of the immunological memory? Already we talked about the primary immune response and the secondary immune response. And also we discovered what the meaning of the memory cells and what the difference between the primary and the secondary immune response. Let's start of the characteristics of the primary and the secondary immune response. Primary immune response and the secondary immune response. Primary immune response is the response upon the encounter of the antigen for the first time. However, the secondary immune response is the encounter of the same antigen for the second time. It's called the secondary immune response as its name indicates. So the experiments of document A, they illustrate the time course of the primary and the secondary immune response against the tetanus toxin. In order to follow this time course, the amount of specific antibody produced and released in the plasma following antigen injection is measured at different time intervals. So in order to see the primary and the secondary immune response after the injection of tetanus toxin, what they are going to do, they are going to do the experiment of injection of the tetanus toxin and observing the amount of the antibodies. Let's start with the first experiment. First experiment, what did they do? They injected the animal with tetanus toxin. Tetanus toxin, as I said before, it's toxic. This leads to the death of the animal, and the amount of antibody was 0 AU, 0 arbitrary units. This means that the tetanus toxin is fatal and it's pathogenic. It causes the death of the animal since there is no secretion of antibodies. However, after the injection in the second experiment of the tetanus toxoid. So tetanus toxoid or tetanus toxin elapse is a weakened one. It's a weakened one. It's a very, very weak. It's like a vaccine and كثير مخفف. So it leads to the production of a memory against this animal. So this tetanus toxoid, it leads after its injection, it leads to the survival of the animal. It leads to its survival. So the survival of this animal, it leads to an increase in the level of antibodies. So the level of antibodies increases about after 1 week. It increases to about 1 AU. This increase in the level of the antibodies, it shows that the immune response is working, is acting, as is functioning uh, against the tetanus toxoid. So we have an immune response against the tetanus toxoid. However, after the injection of the tetanus toxoid 6 months later. So we have injection of tetanus toxoid after 6 months. This this tetanus toxin injected after 6 months, it leads to the survival of the animal. But as you see here, we have increase in the level of the antibodies up to a higher level. So the amount of antibodies, it increases up to a higher level. It increases to about 2 AU after about less than 1 week, after half a week. So this means that when we encounter the antigen for the first time, here when injecting the tetanus toxoid, which has the same antigen as the tetanus toxin, so this leads to the survival, and we encounter the antigen for the first time. So this leads to the increase in the level of the antibodies. However, the, the injection of tetanus toxin after 6 months, so this leads to the encounter for the antigen for the second time. It leads to the survival, and it leads to the increase in the level of the antibodies up to the double, approximately to about 2 AU, while in the primary it was 1 AU. As you see also, it's faster than the primary. It starts after half a week, but here in the primary it starts after 1 week. So it's faster. The delay period is less than, is smaller. However, it protects for a longer period of time. This is what we call it, it's more persistent, while here it protects only for a short period of time. These are the characteristics about the primary and the secondary, and you can refer to the table that we compared between the primary and the secondary immune response. This is the differences in the primary and the secondary immune response. Basis of immunological memory. So when the antigen is encountered in the body, it causes a clonal expansion of specific B and T cells. This clone will give rise, on one hand, to short-lived effector cells, and on the other hand, it will give birth to long-lived memory cells. So this expansion of the B and T cells, the clonal expansion, they increase in number. They give the short-term cells, which are the effector cells, like effector B cells. They are able to kill. They are plasma cells that can secrete antibodies, or the effector T cells, T cytotoxic, that can kill other cells using the enzyme, using the perforin and the granzymes. Or they can give birth to memory cells. These memory cells, they are long-lived memory cells. The reintroduction of the same antigen in the body, it activates memory T and B cells. These memory cells are more differentiated, and they are more numerous than the cells stimulated in the primary immune response. This is the secondary immune response. These are the characteristics that we cover them. It's faster than the primary, it's more amplified. What the meaning of more amplified? It has higher amount of antibody, and it's more persistent, and they can say it's more durable. So it protects for a long duration of time. So this is the characteristic of the secondary immune response. So why the secondary is more persistent, more amplified, and it's faster? Because it contains the memory cells. So the primary بيجهز له memory cells, the secondary. That's why the secondary is going to be faster, it's going to be more amplified, and it's going to be more persistent than the primary due to the presence of the memory cell that play an important role. Importance of immunological memory. A large number of vaccines were developed in the 20th century and marked a turning point in the history of medicine. So, relying on the principle of immunological memory, vaccination has made it possible to win these battles without, without a child having to suffer illness or death. So here it covers. So, for example, before we have measles, we have a disease, is poliomyelitis. We're going to talk about it. All of them, they are fetal diseases sometimes and can kill the baby. The microbial inactivated proteins, it can kill either it kills an individual. So a vaccine, what does the vaccine contain؟ هلا في عندك for example COVID vaccine. The COVID vaccine, it either contains the attenuated. or the killed microbe or the microbial inactivated proteins. So they contain the virus or the bacteria or the killed microbe or attenuated, meaning that it's very weakened or inactivated proteins. So therefore, it retains the microbe's antigen while being harmless, doesn't cause a disease. Immune memory developed following the primary immune response induced by the vaccine provides long-term protection. This is illustrated in document C. We're going to talk about it. Today, there is almost no reason to worry. Smallpox has been totally eradicated since 1980. The poliomyelitis, the شلل الأطفال, infantile paralysis, should follow in 2003, and almost all infectious childhood diseases have become rare in countries where vaccination programs are well implemented, including Lebanon. So poliomyelitis هي شلل الأطفال كان زمان it causes a lot of problems and even bad, and here nowadays it causes nothing due to the vaccination. So here, if you want to move to document C, it shows the results of the epidemiological survey performed in the years between 1930 to 1979. Each arrow here indicates the date of the introduction of the vaccine for each disease. In the two diseases, عندي whooping cough دي الساعة بتيجي مثل بصير يصفر الصوت, and we have poliomyelitis اللي هي شلل الأطفال. Before introducing the vaccine, the rate was about 1 over 1 to 10 over 10,000 inhabitants. So again, about 10,000, 10 over 10,000 inhabitants. So when the poliomyelitis vaccine was introduced at this arrow, لما كان في البوليomyelitis vaccine introduced, the frequency of the disease it decreases to about 0.01 per 10,000 as the vaccination rate increases. So this is for the vaccination rate, and this is for the frequency of the infected individuals. So here, as the vaccination rate increases, the frequency of the disease should decrease. And also here, what happened here? Here we have whooping cough. Before introducing the vaccine, the frequency of the disease was about 100 per 100,000. When we introduce the vaccine at this arrow, the frequency of the disease it decreases below below 10 per 10 or 100,000 as the vaccination rate increases. But we have here an increase again. Why? Because we have increased again, maybe there is a problem in the vaccination schedules or vaccination program for this disease. So some problems with vaccination, بطلوا يعطوا vaccination, they decreases the rate of the vaccination, and this will lead to the increase a little bit for the frequency of the disease, increases the frequency of the disease. So here you have to know that vaccination has an important role in decreasing the effect of the disease. Copy the documents, interpret the results obtained in the experiment of document A, and draw out the characteristics of the secondary immune response. Already we interpreted this. We have to say what is the clinical effect of the injection of tetanus toxin. We said that tetanus toxin it provides immunity against tetanus toxin since it introduces the antigen of the tetanus toxin, but in a weakened way. Justify the characteristics of the secondary immune response in document B. So we have to talk about the characteristics of the secondary immune response in document B. Why do we have secondary immune response? It's more amplified, it's more persistent, and it's, for example, faster. Why? You are going to describe this. And also, we have to move what are the consequences of the introduction of vaccination on the evolution on a number of cases in the diseases presented in document C, and what is the consequences of the decrease in the rate of vaccination of whooping cough after 1975. So let's talk about this step by step. So probing the document question number one, interpret. After the injection of the rabbit of tetanus toxin, it dies, and no antibodies was detected in the blood. So what does this indicate? This indicates that the tetanus toxin is fatal, it's pathogenic due to the absence of the antibodies. After the injection of the rabbit with tetanus toxin, it stays alive. The amount of antibodies was null at the instant of the injection of toxin. It stays null to the beginning of the first week after the injection. where it starts to increase and reaches the maximum 1.2 AU 2 weeks after the injection. Then the amount of antibody decreases to 0 AU after 1 week of reaching the maximum, then remains 0 till the fifth week after the injection. So what does this indicate? This indicates that the injection of the toxoid provokes or it facilitates or it causes the secretion of antibodies. However, 6 months later, this rabbit, the same rabbit, was injected with the tetanus toxin. It stays alive. The amount of antibodies is null at the onset of injection. It takes less time than the primary response to increase, where it starts to increase 3 days after the injection and reaches the maximum 3 AU, which is higher than approximately the double than the one obtained during the primary immune response. On the fifth day of injection, the amount of antibodies starts to decrease slightly, but it remains higher than 2 AU, but it remains protecting the body even after the fifth week of the injection. The antibodies they persist for a long period of time during the secondary response more than during the primary immune response. So what does this indicate? This indicates that the tetanus toxide provides immunity against the tetanus toxin and the secondary immune response is faster. It starts faster. It's more amplified, meaning that it contains high amount of antibodies and it's more persistent. It has long lasting period compared to the primary immune response. Part two, I'm not going to talk about the consequences of the part two. It says about the clinical effect of the injection of tetanus toxoid. Already we talk about this. Moving to part three, during the primary immune response memory cells, they are formed. These cells, they later become effector cells during the secondary immune response. So the number of the effector cells will increase, and that's why the primary immune response it has higher the effector cell than the primary, and for this reason, the secondary is going to be faster, it's going to be more amplified, and going to be more persistent or more durable. Part 4. Before introducing the poliomyelitis vaccine between the years 1935 and 1960, the frequency of the poliomyelitis increases with fluctuation. It fluctuates and the depth of or it increases or decreases between 1 to 10 over 10,000 inhabitants. While after the introduction of the vaccine, this frequency it decreases about 100 times to reach 0.01 per 100,000 of the inhabitants. So here indicate the vaccination decreases the rate of the disease. While moving for the whooping cough, before the introduction of the whooping cough vaccines between 1935 and 1960, the frequency fluctuates between 50 and 100 over 10,000 inhabitants. While after introducing the vaccine in 1960 and increasing the rate of vaccination from 65 to 75 in years 1975, the frequency of the disease decreases to 100 times and it reaches 5 over 110,000 inhabitants during the 1975 year. So what does this indicate? This means that the vaccination it decreases the number of the cases, but the vaccination rate is decreased in 1975. It decreases to about 40%. This decrease in the vaccination rate is followed by the decrease in the frequency. So when we decrease the vaccination rate, قلل vaccination rate, so this will lead to the increase in the frequency of the disease that reached about 50 for 10,000 inhabitants in 1980. So this indicates that the vaccination it decreases the number of cases in the population, and that of the frequency of the disease is inversely proportional to the vaccination rate. Yani as we increase the vaccination rate, the frequency of the disease decreases. As we decrease the vaccination rate, the frequency of the disease will increase. So now we finished document 7 talking about the characteristics of the primary and the secondary immune response. And now we are going to move to document 8 talking about the diagnostic application of the antibody properties. So antibodies, they are highly specific for their antigen. This makes them valuable biological tools. that can be used to detect particular molecules. So in a blood, for example, or urine, or some tissues which contain complex mixtures of different molecules, this specificity of the antibodies makes them possible to detect particular antibodies in individual serum by using the corresponding antigen. So antibody-antigen reaction are widely used. So they use the antibody-antigen reaction to detect the different antigens and to detect the different diseases and so on. So let's see what do they do. Serological test. Serological meaning that they use the serum. The serum meaning that they contain the antibodies. Agglutination reaction. When it's not about agglutination, when we talk about the antigen and antibodies in the blood groups. Each antibody has two antigen binding sites. When antibodies react with the antigen expressed on the cell surface, they form molecular bridge between the cell, causing the agglutination. This is the agglutination that's shown in figure 1 or in tube 1 of document A. Here we have agglutination one here. We have negative test. There is no agglutination in tube 2. The aggregates can be seen with the naked eye. So you can see them في عندي aggregates or you can say the complex or the agglutination using the naked eye. Even though the blood groups, whenever you add blood antibody A to blood group individual A, so antibody A will react with antigen A. So this means we have agglutination. It can be seen by the naked eye, by the way. So the diagnosis for typhoid, for example, relied on the screening of anti-salmonella antibodies in the serum of the patient. So suspension of killed artificially colored salmonella is mixed with the test serum in a tube. After incubation, the suspension is observed. If the bacteria are agglutinated, it means that antibodies they are present in the serum. So the diagnosis is confirmed and they have anti-salmonella antibodies. If the bacteria remain free, it means that the serum does not contain anti-salmonella antibodies. This is shown in document 8. So here it has anti-salmonella antibodies, so it has the typhoid. While 2, it has no anti-salmonella antibodies, so there is no typhoid. So this is the experiment that's used to determine the antibodies. Immunodiffusion in gel. In agar gel that's used in the agar gel, that's used also in the gel electrophoresis, antibodies and antigens they are placed in hollowed wells. يحطوهم بوells مثل فتحات. They can migrate in all directions in this human medium. The contact between an antibody and its specific antigen constitute an immune response that can cause something called a precipitation arc. So this one I need more, which is the agglutination reaction using the antibodies. Immunomarking test. Please have to pay attention in this immunomarking test, which is called the ELISA technique. In the ELISA technique, enzyme linked immunosorbent assay. This technique is called the ELISA, enzyme linked immunosorbent assay. It's the investigated molecule is detected by the sequential addition of two reagents. So we add, it has different steps. We're going to talk about these steps as well. A specific antibody for the molecule, which is coupled to an enzyme and a colorless substrate that is transformed into a colored product upon the contact with the enzyme. So we add specific antibody for the molecule. This antibody is bound to a specific enzyme, and another substrate which is colorless is transformed into colored when it binds to the enzyme, and meaning that when it binds to the antibody. So the steps of the ELISA technique, so human test serum is incubated in a plastic plate or a plastic one that has been covered with HIV antigen that causes the AIDS. If no, if an anti-HIV antibody they are present in the serum, they will bind to the antigens. Other antibodies they remain free. So First of all, human test serum is incubated inside a well. So this is a well. حطينا فيها a human test serum. حطيناها in the plastic well that has been covered with HIV antigen. So في قلبها HIV antigen. If anti-HIV antibody were present, so they bind to the antigen. Other antibodies, they remain free. So the antibodies that are specific for HIV, so anti-HIV antibodies, they will bind. Other, they remain free. So what do we do for the free? The plate is washed abundantly. Only specifically bound antibodies will remain attached. So those that bind to the HIV, they will remain attached. Other they are free, they will be eliminated as shown in this figure. Step number three, an antibody that's specific for the human immunoglobulin and coupled to an enzyme is added to all the well. So antibody that's specific for the human Ig and coupled to an enzyme, so specific antibody coupled to an enzyme is added. After incubation, the plate is washed again. So بنرجع بنغسلها مرة تانية. The enzyme substrate is attached. So this substrate is attached. Its transformation into a colored product, meaning that it has an antibodies indicate the presence of the enzyme bound to the one and that of HIV antibodies in the serum. The infection by the anti-HIV antibodies in the serum is detected. So the above test is a appropriate test or appropriate one. So here we can detect the presence of the antibodies that are found here. Another technique is called the immunofluorescence technique. Immunofluorescence, it's used to detect cellular antigen and not soluble cellular antigens that are found on the surface of the cell. Immunofluorescence, as its name indicates, we have fluorescence. Cells to be tested are fixed on a glass slide. They are incubated with a specific antibody that is chemically bounded or coupled to fluorescent substances. The antibody binds to the cell only in the presence of antigen. When the cells are later washed and they are submitted to UV ultraviolet light beam, the fluorescent substance emits a colored light that can be observed with a specific microscope, which is called the fluorescence microscope. So, starting with the first one, what is the agglutination technique described in the previous documents? Schematize the mechanism relative to the formation of the precipitating arc. Referring to document C, draw a similar diagram in case where the tested individual is seronegative. What the meaning of seronegative? It doesn't contain the anti-HIV antibodies. This meaning of seronegative. Seropositive, it contains schematically illustrate the different phases in the immunofluorescence test. This agglutination technique that has previously described is the agglutination of the red blood cells, hem agglutination, that may help to determine blood groups of the samples. Two, schematize the formation of precipitation arc. The binding of the antigen to the antibody leads to the formation of the precipitation arc. Moving to three, a schematic interpretation of the ELISA test in the case where it's seronegative. Seronegative, so the antibodies in stage one will not bind to the antigen of the HIV, even though we added the enzyme bound to the substrate and then we wash it. So all of them they are removed, so no binding. Where here we have the binding of the antibodies to specific antigen. While in the seronegative, there is no binding and the antigen they remain free and the antibodies they are removed upon washing them. Stages of the ELISA technique. So these stages you have to understand them, not to study them by heart, but you have to know the steps. Test serum is incubated in a plastic plate or well that is well covered with HIV antigen. For example, here we took HIV. Since there is no anti-HIV antibodies in the serum, so no antibody will bind. This is the case of seronegative, by the way. Okay, this is the case of the seronegative. While in the book, this is the case of seropositive. So no antibodies will be in the serum. So no antibody will bind to HIV antibodies. So HIV antigen, sorry. So all of them will be free. Stage two, the plate is washed. All of the free antibodies will be eliminated. Stage three, a human anti-Ig antibody coupled to an enzyme So antibody coupled to an enzyme is added in the well. Since there is no human Ig in the well, the newly added antibodies remain free. So after incubation, the plate is washed again. Stage 4, the enzyme substrate is added. Since there is no binding, there is no enzyme, so the substrate remains clear and there is no change in the color. Okay, while for the schematic interpretation of the immunofluorescence technique, so we added the antibodies, they bound, so the binding of these antibodies will lead to the uh, to the binding of the fluorescent substance, and the fluorescent substance will be under the UV beam and will cause the appearance of the fluorescence under the specific microscope. So the cells to be tested, they are fixed on a slide and they are incubated with the specific antibody coupled with a fluorescent substance. Stage 2, the slide is washed and then the free marked antibodies are eliminated. Stage 3, the cells that are subject to an UV light, and then the fluorescent substance will emit a colored light that can be observed under a fluorescence microscope. Okay? So here note you have to know that agglutination is the reaction between an antibody and a cellular antigen, whereas precipitation is the reaction between an antibody and a soluble antigen. Next time we're going to move to the exercises on the book.